We're happy to have you join us today. My name is Lourdes. I am Director of Patient Services with the Cholangiocarcinoma Foundation. Um, I would like to introduce you to our speaker today. Dr. Mark Yarchoan is an Associate Professor of the Medical Oncology at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins in Baltimore, Maryland. He has a clinical focus on the treatment of hepatobiliary cancers, and he runs an NCI-funded laboratory focused on developing novel immunotherapies for hepatobiliary cancers with a focus on neoantigen-targeted vaccines. I would like to remind everyone to please enter any questions into the Q&A and or chat box. Please make your questions general in nature, as Dr. Yarchoan cannot answer very specific questions. And I'm going to hand it over to you.
Hey everyone, good afternoon. I'm Mark Yarchoan, and it's a real pleasure to speak to all of you. I know I've met many of you at the CCF meetings, and for those of you I haven't met, it's really nice to chat today. I'm really speaking on behalf of our team here at Hopkins, so again, everything that I'm going to present, there's a large team of people that contributed to everything, and in particular, the trial that I'm speaking about is co-led by my colleagues Marina Baretti and Nilo Azad here at Hopkins. So I'm going to give a science lecture, and then we'll get to the clinical trial that we're really excited to open very shortly here at Hopkins. But I want to explain what it is that a vaccine does and why we're excited about the trial that we're hoping to open very soon. So this is maybe new information for some people, but our body has an amazing ability to fight cancer. And the idea of a cancer vaccine is not at all new. In fact, it's something that people have been working on really since the 1940s. It's just that we haven't had the best tools to make a cancer vaccine until recently. And it's now is the golden era of cancer vaccines because we've learned so much, in part because of the COVID pandemic, and a lot of the vaccine platforms that are now being moved into cancer were actually developed rapidly as a result of COVID. So there is something good that came out of this horrible pandemic that we had. So I want to go back to the 1940s to explain a very simple science experiment that was done a long time ago. So you take a mouse and you put some cancer cells into a syringe and you inject those cancer cells into the mouse, and lo and behold, a cancer grows. And then you cut the cancer out. So the mouse now had surgery and is cancer-free. It's been known now for many decades that if you take that mouse and you re-challenge the mouse with the exact same cancer again, from the same syringe, injected the same way, the same number of tumor cells, they no longer grow. And so this very, very simple experiment that's been reproduced thousands of times by thousands of groups, I think convinces everybody that the immune system has the ability to recognize cancer and actually be vaccinated against cancer. We just haven't really figured out how to do this. Now, I just want to say there are cancer vaccines that are widely used, but they are cancer prevention vaccines that work against viruses. So just for example, we have the HPV vaccine that protects against HPV virus, which leads to cervical cancer. That vaccine prevents about 1% of adult human cancers caused by the HPV virus. The hepatitis B virus also can be vaccinated against and is a major cause of liver cancer around the world. But these aren't true cancer vaccines. They're vaccines against viruses that cause cancer. So what I'm going to be talking about today is vaccines that actually target cancer itself, and how these vaccines work and what we've learned over several decades of failure that has led to a lot of excitement in the field very recently. This, again, I apologize, but this is going to start out as an immunology lecture. So you're back in college today. So this right here is a cancer cell on the left. And every cell in our body is constantly showing its contents to the immune system. And it's one way that our body constantly fights intruders, is when we get infected with a virus, the virus infects a cell, The cell shows the immune system that it's infected by a virus through the system that we have, the HLA system, and our body's immune system can destroy the cell that's holding the virus. It's how we get rid of a lot of pathogens in our body. And it turns out that little pieces of cancer are also presented to the immune system through the same system. And that is how our body's immune system can see cancer cells. Now, some of you may be aware of this, but cancers contain mutations that make them a cancer. So cancer is a cancer because a normal cell picked up a mutation. That mutation made it grow. That mutation, even though it gives the cancer an advantage, it also gives the cancer a vulnerability, which is that mutation codes for an abnormal protein that our immune system can see. And so one of the things that we published a long time ago that a lot of groups have also published is that the more mutations a cancer has, the more likely our current immunotherapies are to work. And so the cancers that immunotherapy works really well in, cancers like melanoma and colorectal cancer with mismatch repair deficiency, have lots and lots and lots of mutations, and therefore are very likely for the immune system to see and recognize those cancers. Unfortunately, cholangiocarcinoma is one of the cancer types that is less responsive to immunotherapy, in part because most cholangiocarcinomas have a relatively low tumor mutational burden. Very few mutations, very few abnormal proteins, less likely for the immune system to see those abnormal proteins and attack cholangiocarcinoma. So the idea behind a cancer vaccine is pretty simple. If the cancers that have lots of mutations and lots of abnormal proteins are the ones that the immune system can attack most easily, then a vaccine that makes the immune system see those abnormal proteins should make cancer immunotherapy work better. And so that's the basic premise behind cancer vaccines that are in development today. They target those abnormal proteins that cancer cells have. They help the immune system see those abnormal proteins. Now, I want to just rush through a couple of very exciting studies that have happened recently in other cancer types, not cholangiocarcinoma. But as people focused on a rare cancer, I think we can learn a lot from other tumor types. So this hit the lay press and was a topic of a great excitement. There was a trial of a vaccine for patients with pancreas cancer. So patients with pancreas cancer underwent surgery. We know that patients with pancreas cancer have an even higher chance of that cancer coming back after surgery than even cholangiocarcinoma. This group, led by Vinod Balachandran at Memorial Sloan Kettering, sequenced the cancers that had been removed, the pancreas cancers that had been removed, and gave a cancer vaccine that was personalized for that patient, trying to convince the immune system to see the antigens in that one patient's cancer. So it's sort of incredible that this is possible. This is the Human Genome Project for a single patient. We sequence all the mutations in that tumor. We make a single vaccine for one patient. And what the group showed is that about half of patients who got their personalized vaccine made an immune response to it. But the 50% that had an immune response were much less likely to have their cancer come back than the patients who didn't have a response. And that's shown here in the waterfall plot. In fact, none of the patients who mounted a response to their personalized vaccine had their cancer come back. If you didn't mount a response against the vaccine, your cancer was very likely to come back, usually in less than a year. And so it provided us some initial evidence in a very tough tumor type, similar to cholangiocarcinoma, that vaccines, when they mount a response against cancer, can be meaningful. We led a very similar trial in liver cancer, but for patients with HCC, the other form of liver cancer. This was led by our group here at Hopkins. What we showed is when we combine a vaccine with a checkpoint inhibitor, in this case pembrolizumab, about a third of patients had major tumor shrinkage in a tumor type where checkpoint inhibitors normally don't work by themselves. So again, adding evidence that these vaccines are actually starting to work for the first time. And then perhaps most convincingly, This is a trial in melanoma, which is the most immunogenic tumor type around, where patients with melanoma had surgery to cut out their melanoma and then got randomized to a vaccine or no vaccine. In this case, this is led by Moderna, household name after COVID. They made a personalized vaccine for each patient. The patients who had the vaccine were less likely to have their melanoma come back than the patients who just got pembrolizumab without the vaccine. This has gone into a confirmatory phase 3 trial, but actually these results are so striking with about a 50% reduction in the cancer coming back that the FDA looks likely to actually give this vaccine an accelerated approval for melanoma. Now, those of us who treat cholangiocarcinoma, we often look at our colleagues in melanoma with a little bit of envy because a lot of things happen first in melanoma. So we had the approval of a checkpoint inhibitor just 2 years ago. They had it back in 2011. But we're looking to cut the time down and not just do things a decade later for cholangiocarcinoma. And so I'm very hopeful that that's going to be the case. So I'm going to skip over something and just talk very quickly about how our vaccine works and the approach that we're taking. So we know that the abnormal proteins in a cancer cell are targetable with a vaccine. One of the things that's kind of interesting about cholangiocarcinoma, as many people may know, is that there aren't that many mutations in cholangiocarcinoma. And the mutations that you find actually tend to be very common mutations in patients with cholangio. So, for example, a lot of people with cholangiocarcinoma have the same mutation, an IDH 132C mutation, which means maybe a single vaccine could actually be used to treat not just one person with cholangiocarcinoma, but a lot of patients with cholangiocarcinoma. And so that's the basic idea behind our vaccine is it's a semi-personalized vaccine. So whereas Moderna and BioNTech are sequencing one tumor and making an N of 1 vaccine for that patient, we are going after mutations that are shared from patient to patient to patient with cholangiocarcinoma. And we think that there's some reasons why this is a really good approach. The first one is we can make a vaccine very quickly because we have vaccines that already cover a lot of patients. So you walk in the door, you know that you have a KRAS G12D mutation. We have a vaccine that already is prepared for patients with KRAS G12D. But also, because these mutations actually drive the cancer, if we can make the immune system see the drivers of the cancer, we think we can get a much deeper response than if we just go after whatever we find with sequencing. So this is a map of some of the common mutations that we find in patients with cholangiocarcinoma. And our vaccine covers many of these mutations, and I'll show you the list of mutations. So patients who have a common cholangiocarcinoma mutation might potentially be a candidate for our trial. This is how the trial works. So essentially patients with cholangiocarcinoma who have already had at least one dose of gemcitabine cisplatin and durvalumab, or gemcitabine cisplatin and pembrolizumab, the two sort of preferred frontline therapies for cholangiocarcinoma, are eligible for this trial. And patients who go on our trial receive two different checkpoint inhibitors. One is durvalumab, which is, again, the standard drug used for cholangiocarcinoma. We give it with a second checkpoint inhibitor called tremelimumab, and that is an anti-CTLA-4 therapy that might prime the immune response a little bit better, so it gives a little bit stronger of a boost of immune activation. And we give it with our special vaccine, and this is the schedule here. Unfortunately, this vaccine is only going to be available at Hopkins because it's actually physically made here and it's not very stable. So it's not something that we can just ship around the country. So patients do, unfortunately, need to be here for treatment. The vaccine is given once a week at the beginning, but then becomes spaced out. So eventually it's an every 4 weeks treatment. So which mutations are covered by our vaccine? This is the list of mutations covered by our vaccine. Um, I apologize if patients do not have a mutation on this list. We certainly hope to expand the list of mutations that we're covering, but due to limited funds, these are the most common mutations in cholangio and the ones that we could afford. And essentially, we cover all the most common KRAS mutations, IDH1 mutations, IDH2 mutations, BRAF V600E, and two PIK3CA mutations, and a rare FGFR2 fusion event. I'm sure there are patients who are on this call wondering, do I have one of these mutations? And I would just say many patients with cholangiocarcinoma have molecular testing, for example, Caris or Tempus or Foundation Medicine or a company like that, where the mutations in the cancer have already been sequenced. Those reports will say the specific mutations present in the cancer, and patients can very easily look to see if they have one of the mutations on this list. And if patients do have one, then potentially this trial could be an option. If not, I don't want you to lose hope. There are other vaccine trials coming. Some of them will have a broader list. Some of them will be personalized for an individual patient. And we certainly are hoping to expand the list of vaccines that we're offering here at Hopkins. This is just an initial menu at this time. And obviously, we want to make sure that patients are well enough for an experimental therapy. And so we want to make sure that they have good numbers of blood cells, in particular the white blood cells that actually respond to vaccines, and adequate heart and liver function. As many of you know, I spend a lot of my life in a laboratory trying to come up with new treatments for cholangiocarcinoma. And one of the things that I'm most excited about with this trial is, I think all of us who live and breathe cholangiocarcinoma all day are committed to coming up with a cure for this cancer. And patients who participate in this vaccine, we certainly hope the vaccine helps them. But the one thing that we can guarantee is that it will help other patients. And in particular, in this trial, what we're hoping to do is for patients who have a good response to the vaccine, if their cancer shrinks, we know their immune system responded to the vaccine and actually fought the cancer. And so what we're going to do is take the immune cells from the patients who do well with the vaccine and actually find the T cell receptors, the immune cells that are fighting their cancer. And so their immune cells can actually become the drug for the next patients. And so what we hope to offer in a couple of years is a patient who comes to us with just, for example, an IDH 132C mutation. We can say, hey, you're welcome to join our vaccine study, or we can take a shortcut and we can use the immune cells from a patient who did really well with the vaccine trial, and we can put those immune cells directly into you. And so we do ask for patients who are participating in our trial to allow us to take blood, not only to monitor their immune responses, but also to become a drug for future patients. So we thank all patients who participate in research. And I think that that's going to be key to moving forward for this cancer. That was a very, very rapid immunology lecture and then a brief introduction to our trial. And I'm happy to take questions. And oh my gosh, there are so many questions already in the trial in the chat. So
the first one that came in through email was just if there were any approved immunotherapy that could work with the vaccine. And I don't know if you have an answer to that.
Yeah, so durvalumab and pembrolizumab are two checkpoint inhibitors. They broadly take the brakes off the immune system, and they're both approved for patients with cholangiocarcinoma. Unfortunately, I think there's a lot of room for improvement. So we know that only about 5 to 10% of patients actually respond to a checkpoint inhibitor alone in cholangiocarcinoma. We are including durvalumab as part of our treatment paradigm for this trial. But again, what we're trying to do is not just turn the immune system on, but actually direct it against the cancer. And that's what we hope that the vaccine does.
Okay. Are patients that had prior CTLA-4 plus PD-L1 still eligible?
To my knowledge, they are, yes.
Okay. For everyone on the call, yes, this is being recorded and will be on the YouTube channel 24 hours after we end this webinar. Are all FGFR2 fusions eligible or only FGFR2 INA?
At this time, only FGFR2 INA. We think that that's a very immunogenic particular fusion. We do hope to have others in the future.
Um, will vaccines work for individuals who have no known mutations or some they cannot identify?
Um, to make a vaccine, you need to have something to target. And again, our vaccine will only work for the targets that we have on that initial list. Um, I don't know if this person who asked the question um, wants to share more, but cancers are cancers because they have a mutation. That's our understanding of what a cancer is. And if a patient comes to my clinic with a report that says they have no mutations, I usually tell them that there's a mutation there. We haven't looked hard enough for it. And so for all patients, I encourage you, first of all, to make sure you have whole exome RNA sequencing. Some platforms, like, for example, an old version of Foundation Medicine did not look at RNA, and many cholangiocarcinomas have abnormal splicing that can only be picked up at the RNA level, not the DNA level. So I would just say talk to your oncologist, but if you really are walking around with a piece of paper that says, I don't have any mutations in my cancer, chances are that more testing will help find them.
Can a patient who has been getting durvalumab for 2 years apply for this clinical trial?
Yes. So patients can have had any number of prior therapies, and prior durvalumab is totally fine. And actually, we do know that vaccines, when they work, they take a while to work. It's not like chemo where you give a dose of chemo and then immediately the tumor starts shrinking. The body has to see the vaccine, respond to the vaccine, grow up immune cells, and then those immune cells have to fight the cancer. So actually, the optimal patients, I think, for this trial are patients with generally lower disease burdens who can actually wait a couple weeks for a response. So someone doing well with durvalumab maintenance who maybe has just very slight tumor growth where the idea of switching to something else makes sense, but there's no emergency. Those are the kinds of patients that I think would be best suited for this trial.
Okay. What if someone received Gemcis but not durvalumab? Are they also eligible?
They should be eligible, yep.
Okay. What are the most likely risks of this particular treatment?
Yeah, I mean, I think it's important to emphasize that this is a brand new treatment. And, every time that we bring something into patients for the first time, there's always the risk of a terrible side effect that we were not anticipating. And, I say this to all my patients. All of you are so brave to participate in new things. That said, our best drugs of tomorrow are all going to be in trials today. So, I think we need to balance risk and potential benefit. Um, I think the checkpoint inhibitors that are used as the backbones, the durvalumab and the tremelimumab, have very well characterized risks in patients, including in cholangiocarcinoma. The major risk is autoimmunity, so the immune system can attack itself instead of the cancer. For example, a rash if you attack your skin, diarrhea if you attack your bowels, shortness of breath if you attack your lungs. The risk of that is on the order of about 10 to 20% in most studies of the two checkpoint inhibitors. And most of the time, if we give steroids, we can calm the immune system down and reverse that. I have to say the track record of cancer vaccines so far has been very encouraging in terms of safety. We certainly expect to see some reactions at the time of the vaccine administration. Patients can get a welt, a fever, headaches, things that people report after getting, for example, a COVID vaccine. But in general, cancer vaccines have been very safe. And in general, in other trials, we have not seen terrible toxicities from cancer vaccines. That said, I think it's important to just reiterate, we just don't know because this is a brand new trial.
Okay. Is your plan to use the vaccine after surgery to prevent disease recurrence?
So our trial is written very broadly, so patients can or cannot have measurable disease. Again, this is really to try to figure out, can we get immune responses against the vaccinated peptides? And so, I think patients who have micrometastatic disease, so patients who have a little bit of disease after surgery, or maybe no disease but are at very high risk of recurrence, we're certainly happy to talk to those patients about whether the trial makes sense.
Must there be a certain disease burden to be eligible, for example, tumors versus lymph node only?
No, patients do not need measurable disease. That said, patients with measurable disease are certainly welcome.
Okay. Does this help both intra and extrahepatic cholangiocarcinoma?
Yes.
Okay. Um, when do you expect to launch this trial, and how long will that trial last?
Yeah, so we are, I believe, about 8 weeks away from formally opening our trial. Um, that said, for the first couple of patients, we can't start everybody all at once in case there's an unexpected side effect. So for the first couple of patients, we need to dose them spaced out by a couple of weeks. And we already have a couple of patients who are waiting. So I think for patients on this call, we're probably about 3 months away from formally being able to offer this to everybody.
Okay, for anyone who's interested in this trial, who should they contact?
Um, That is a great question. So we do have a clinical trials referral office. I don't know if I should send an email with that contact info and maybe it could be added in some way to the website.
Sure, we can definitely add that to the recording, whatever that is. Absolutely. And I'm
That's
sure
great.
that once the trials open, we can also, the foundation can post it on the clinical trials page on the website as well.
Yeah. We're not that hard to find. My last name is pretty unusual, so you can you can just email me.
Does it matter if the mutation is germline or somatic?
No, I mean, most of these mutations are going to be somatic. It's a very good question, whoever asked it. But in general, a lot of these mutations would not be consistent with life if they were in every cell in our body. So, um, but as long as they're in the cancer, they could be targeted.
Okay. What about a patient who has FGFR3 and IDH? Are they eligible, or maybe one day will be eligible?
Yeah, so as long as patients have any of the mutations on that list I shared, they're potentially eligible. So we do not have the FGFR3 included in our vaccine, but we do have IDH1. So if the patient has both, that's fine. They just need one. Um, so that patient should be eligible.
Okay. Does the patient need to get NGS sequencing before applying for the trial, or will the trial do that for them?
Um, I would be happy to see the patient in clinic and order NGS testing for that patient. That said, every cholangiocarcinoma patient, I can't emphasize this enough, should have NGS. So I don't want you to wait 3 months and come here to see me and ask me to order NGS. Go to your oncologist today and ask for NGS testing. Everybody should have this done. It's approved, it's covered by insurance. And again, I think that this should be done as standard of care for all patients. Obviously, if you come to Baltimore to see me and you don't have it done, I will gladly get it done for you.
Is this only for people with active cancer, or is it also given to patients who are NED?
Um, so we don't explicitly say in our trial that you need to have active cancer. That said, um, I think we need to be thoughtful about not exposing patients who may be completely cured already to the risk of an experimental therapy. So I would say a patient who's had surgery who's at high risk for recurrence, for example, maybe ctDNA is positive or there's something that's really worrying us. Those patients, I think, this trial makes a lot of sense for. Someone who had curative surgery 2 years ago and is doing great, I don't think I would personally recommend this trial.
Can international patients um, work with Johns Hopkins to be included in the trial?
Um, so patients who are international, we don't exclude them from the trial, but the trial does not cover the standard of care aspects of being a patient. And often, if you're international without US healthcare, it can be very expensive to get care in the United States. There are other trials going on in Europe, and BioNTech is opening a personalized vaccine trial in Europe. So there may be other options for international patients besides coming here. Again, happy to refer patients to the international office and they can figure out what the cost would be.
Okay. What are the exclusion criteria apart from the organ functions mentioned? For example, hepatitis B is an exclusion criteria.
I can pull up the criteria and show them all, but hepatitis B is allowed, I believe.
Okay.
Let me see if I can pull it up, but that's one that I remember. Hepatitis B is okay, as long as it's controlled.
Okay. Um, how long would a patient need to stay at Johns Hopkins during treatment, or how frequently would they have to travel?
Yeah, so great question. So the trial is again, the vaccine is given once a week at the beginning for 4 weeks. The treatment itself is very quick. I mean, the vaccine is really just a shot. The patients do need to get the Durva and Tremi once a month. That takes for the Durva alone, it takes about an hour. So but there's obviously time associated with getting checked in and labs and all that sort of stuff. So I would say it ends up being a visit every week for probably in total a couple of hours, and then every 4 weeks after that.
Okay. Is there a specific list of therapies which would preclude eligibility?
So no prior therapies that preclude eligibility. We have any line of therapy. Patients can have any number of prior therapies. And the only requirement is that patients have to have had at least one dose of GEM-CIS and a checkpoint inhibitor.
Okay. Um, what is the difference between this study? Oh gosh, hold on. And the trials that are injecting viruses into the tumor.
Um, so, I think there's many different people working on the same problem, which is how do we make the immune system um, see things that it doesn't necessarily see automatically. A vaccine is one way to do that. You give the body a danger signal plus the thing you want it to see, and it can be educated to see that thing. I think another avenue of related research is turning the cancer itself into a vaccine or an in situ vaccine. And one way to do that is through an oncolytic virus, a virus that is inside of the cancer. And so the body attacks the virus and then starts attacking the cancer. These are two different ways to try to achieve the same result. And I think both of them are very promising. Um, I think in general, I encourage patients to participate in research. I think it's the way that we move forward as a field. And I think given the limited standard options for many patients, it's also an opportunity to have another shot on goal. And I think oncolytic viruses are very exciting. I do want to just make a quick correction. So we do limit the number of prior experimental immunotherapy agents besides anti-PD-1 therapy. So I apologize that I misspoke. So any number of lines of chemotherapy, targeted therapy are all fine. But we exclude patients who've had prior IL-2, interferon, OX40, some other experimental therapies, just in case there's some unexpected delayed immune effects when we give our therapy.
Okay. Is a potential cytokine storm a possible adverse effect of this vaccine?
Because it's a brand new therapy, anything is a possible abnormal side effect, but that's not something that I would expect to see.
Okay. Are there any plans to target BAP1 mutations?
Um, uh, so, right now, uh, this was really funded by a patient donation, and uh, we were able to manufacture the peptides that I showed. Really, the thing that limits us is money, and certainly we hope that over time, we're writing grants and things, and we certainly hope to expand the list. And BAP1 is one of the very common cholangio mutations, and something that I think is imminently targetable as well.
Okay. Would stage 4 patients that have had targeted treatment and scanned as NAD for 6 months qualify? I think you answered your thoughts on that.
Yes, absolutely.
How is the drug administered? Intratumoral or IV?
Yeah, so the vaccine is given as an injection, so very similar to the way that a COVID vaccine or flu vaccine is given. The only difference is we like to actually inject in 3 places at once because we think we activate more immune populations by giving it in both arms and a leg. So it's 3 shots instead of 1. But it's given as a shot. The durvalumab and tremelimumab are just IVs, so they're given through an IV, an infusion.
Okay. Is there a clinical trial number yet?
Very shortly. I think we're about 2 weeks away.
Okay. Is this much different than the Vaccinia CF33?
I have to acknowledge I'm not familiar with that, so I don't know. I'm sorry.
Um, do you cover any travel expenses to be in the trial?
Unfortunately, we do not.
Okay.
I will say that there are organizations, this is just in general for cholangio patients, there are actually organizations that will help patients with travel. Um, Young patients, there's more funding, but adult patients, there's actually um, some flight organizations that will fly patients for trials. So a little Googling and you can find some things.
Yes, and we also have a resource hub page on the Cholangiocarcinoma Foundation website, which lists a bunch of different organizations with different things that they might help out with too. So, are steroids an exclusion?
So patients with active autoimmune disease on steroids are excluded, and that's again because we don't think that the therapy would help them. We need the immune system to be active, and steroids really suppress the immune system. So those patients would be excluded.
How many total patients are you recruiting for this trial?
Um, that's a good question. So right now, initially, we're targeting about 20 patients. Uh, we have a history of expanding our trials when we get more resources, so we hope to offer this to more patients.
Okay. Has this specific vaccine been used already in human trials, and if so, which solid tumor cancers? And if you can share the overall survival rate.
So this is a brand new vaccine. It's never been used before in patients. That said, the basic components of this vaccine have been used before. So this, vaccine contains 2 parts. It contains an antigen and an adjuvant. The antigen in this case is a peptide, a little tiny piece of abnormal protein. And then the adjuvant that we use is called poly-ICLC or Hiltonol. Many other peptide vaccines with Hiltonol as an adjuvant have gone into patients. So even though this is the first time that this is going into cholangio patients and the first time that it's being targeted for IDH1 and some other mutations, the basic construct of this vaccine has been used in many thousands of patients, has a pretty good track record for safety. Here at Hopkins, we have this as a trial for fibrolamellar HCC, which is another rare form of liver cancer. And so we feel very comfortable with the safety profile of these agents.
Okay. Can you share the total length of treatment?
Yeah, so patients can basically stay on this as long as it's working and they're not having bad side effects for it. At 2 years, the trial technically stops if they get to 2 years. But by that point, we think that the benefit of the vaccine has probably been locked in. So, that would be a great problem if people get to 2 years.
Okay. How long do I need to be off any current treatments before entering into this trial?
I believe it is 3 weeks.
Okay.
It could be 4 weeks, could be 2 weeks. I think it's 3 weeks.
Okay.
I can double check if you want. Let's see. 4 weeks. Sorry.
4 weeks. Okay, perfect. Can the vaccine be used in a patient that has an intrahepatic pump? So the HAI pump?
Yeah, so patients who have a history of the HAI pump are certainly welcome to participate, but patients can't be getting active chemo through their pump while they participate in this trial.
Okay. What is the difference between T cell therapy and this vaccine?
The term T cell therapy is pretty vague, but I'm guessing that T cell therapy means TCR therapies. Um, I would say that they're a little different. The goal of the vaccine is to make TCRs in a patient that fight cancer. TCR therapy is a shortcut, so you just get the T cells directly into your body that the vaccine would have made. And I alluded to this, but we certainly hope to generate TCR therapies for future patients from the patients participating in our vaccine trial.
Okay. If a patient is a candidate for liver transplant, can they use the vaccine after the transplant to prevent recurrence?
So the vaccine itself would probably be safe in that situation, but we give the vaccine with checkpoint inhibitors, durvalumab and tremelimumab, and those would likely cause the body to reject the transplanted liver. So unfortunately, patients who have a solid organ transplant are excluded from the study.
Okay. Will some individuals get a placebo?
There is no placebo on this trial, so everybody gets a sort of custom vaccine for their mutations plus durva plus tremi.
Okay. Would being on Tibsovo exclude participation in the trial?
So prior Tibsovo would be A-OK, but again, Tibsovo would be stopped when patients start the trial.
Okay.
And by the way, Tibsovo is an IDH1 inhibitor, as people know, so that's one of the mutations that we certainly hope to target in our trial.
Mhm. If someone has none of the mutations listed but have PDL1 expression, would they be eligible?
Unfortunately, not for this trial, but PDL1 expression means there's some immune inflammation, and maybe a different trial would be a good one.
Okay. Um, Someone mentioned that personalized vaccine in combination with histotripsy can have benefits as there are more circulating cells for the immune system to recognize. Do you have any thoughts on that?
Yeah, there's a lot of buzz about histotripsy. I think the buzz is probably ahead of the science. We don't know if smashing a bunch of cancer is activating or profoundly immunosuppressive. And, I think we need more science. It's really a lot of marketing right now, a lot of buzz.
Do you foresee a vaccine without durva being a replacement for adjuvant chemo following successful resection surgery?
Yeah, that's a great question. I think We generally feel that vaccines work best the earlier that they're used. And so just the way, a patient who um, has active COVID, we don't run and give them a COVID vaccine when they come into the ER with COVID. Vaccines work best in minimal disease or even cancer prevention. So certainly, we want to show that the vaccine is safe. Hopefully, we're going to see that it helps fight cancer. But our long-term goal is to move this as early as possible. And I think in the adjuvant setting would be ideal.
Okay. If someone failed durvalumab, does that exclude them from this particular trial?
It does not. No.
Um, does this trial favor intrahepatic or extrahepatic cholangiocarcinoma?
Either are eligible.
Okay.
And the mutations we're targeting, many of them are found in either extrahepatic or intrahepatic.
You mentioned a similar study by BioNTech for international patients. How does this study and their study differ?
I don't know the details on their study, but I do know that those of us who have been studying cancer vaccines for a long time, it's really nice to see finally some vaccines starting to actually work and a lot of enthusiasm. And I think we're excited about our trial, but I think there are going to be many vaccine trials coming for many cancers all around the globe. So if our trial is not a fit, I encourage you to look around because there may be other trials. Moderna will be launching personalized vaccine trials for many tumor types. BioNTech is as well. And so stay tuned. A lot of reasons to be hopeful.
Is someone with gallbladder cancer eligible for this particular trial?
Yes.
Okay. Um, I know there's questions about if this is only at Johns Hopkins, and yes, it is only done at Johns Hopkins. And if patients are curious, if there are any other clinical trials for cholangiocarcinoma at Johns Hopkins, is there a page that they could go on the website to go and see what those different clinical trials might be?
Um, Unfortunately, we don't. The trials come and go so quickly. I do think clinicaltrials.gov is a great way to look around, and there are other sites that pull information from clinicaltrials.gov. But we're happy to see you and discuss our trials anytime. We're a nice group here. It's not just me; it's my colleague Marina Baretti and Nilo Azad. The three of us really lead the cholangio program here, and all of us are happy to chat.
Perfect. Mm. If a patient has more than one mutation, so for example, ARBB2 high and the PIK3CA, could one of those be selected in that instance to participate?
Yes, and we include as many mutations as we can in our vaccine. So if a patient happened to have, for example, both a KRAS G12D and a IDH1 mutation, we would actually put both into the vaccine for that patient. So you need one to participate, but the more the merrier.
Okay. You had mentioned that sometimes patients, it will take a little bit of time before they see a response with the vaccine. So how long would you wait to take another scan?
Yeah, the first scan happens just around 9 weeks on therapy. And again, we don't necessarily expect a response that early, but we just want to make sure that things are at least controlled and not moving in the wrong direction.
I think we answered a lot of these questions. Can you differentiate between pseudoprogression and actual progression?
Pseudoprogression is one of these things we talk a lot about, but don't actually see that often in clinical practice. I get pretty nervous keeping people on trials if the scans are going the wrong way, especially if they have other options. So, no, we can't really distinguish between the two. I mean, unless there's some dramatic change in ctDNA or tumor markers. But generally, if the scan looks worse, we generally tend to assume that it's real progression.
Okay. Would MSI high patients that have a mutation listed be eligible if prior PD-L1 checkpoint failed? I think you mentioned that.
Yes, those patients would absolutely be eligible. That said, I think those patients may be sensitive to many immunotherapies, and there may be a lot of good options for those patients.
Okay. If there's progression at the 9-week scan, is the trial discontinued?
Patients are allowed to continue past one progression event as long as the treating oncologist feels that it's reasonable to stay on with a close interval scan. At the
Okay.
second progression event, patients cannot continue.
Okay. Will you use CA19-9 tumor marker to measure the success of treatment?
So that's one of the biomarkers that we follow. We'll be following tumor-free, cell-free DNA as well, which can be another helpful adjuvant or adjunct to sort of get a sense of how things are going.
Assuming positive signals in this phase, how will adding more targets look? Will those new targets need to progress through phase 1?
Um, we really think about our program here as being an immunotherapy program, and we're always looking to make it better. So we certainly hope that this becomes an iterative vaccine. We have some new things we're working on in the laboratory, new ways to make our vaccine a little more potent. And so, I really hope that if I'm welcome back here again, love to give updates about how our vaccine changes over time. We hope to add additional targets. We actually hope to make our vaccine more potent by making some modifications to our peptides to make them even more immunogenic. There's an active effort in my lab to do that and others here. So stay tuned. A lot of reasons to be hopeful.
And we will definitely welcome you back to give updates. I know that the community really likes updates, so definitely look for our emails.
Okay.
I think a lot of these questions are very specific to certain mutations being eligible or not, but I know that you said that you're hopeful to expand and include other mutations. So I think that helps to answer those questions.
Yeah, I mean, would it help if I drop the mutations into the chat so people can see it?
Or if you wanted to show that
That chart again?
page, yeah, that would be
Or I can
good.
just put it up and patients can take a look here.
Perfect. So these are the mutations that are eligible for participation in this trial. If anyone wants to see those. And I think we got through all of these questions. Thank you so very much. I know we
Great.
had a few repeats. And
Well,
thank you to
thank you
everyone who
all for,
participated today.
yeah, thank you for your participation and great questions.
Thank you. Appreciate your time. Thank you, everyone.
Thank you.